So our next talk will be from Priya Prakash from Purdue University. The title is, "What do microglia make when they eat amyloid beta?" All right, you have the screen up and we're good to go. All right.
Okay, can you hear me?
Yes.
All right. So hello, everyone. Thank you to the organizers for having me here, and thank you all for being here. My name is Priya, and I'm a grad student in Gaurav Chopra's lab in the chemistry department at Purdue. And I'm excited to talk about some of the new chemical tools and techniques that we have developed in the lab to study glial cell biology, and specifically my work focused on microglial response to amyloid beta. So it is now evident that microglia they have many fundamental roles in the brain. They actively interact with neuronal synapses, and they prune the synapses using complementary proteins and receptors, as shown by Beth Stevens and others in the field. Reactive microglia also secrete cytokines like IL-1 alpha and TNF alpha that in turn influence astrocyte cell state and neuronal health. In a tumor microenvironment, microglia also interact with glioma cells using eat me and don't eat me signals. And more specifically, microglia phagocytose misfolded protein deposits from their microenvironment. Proteins like amyloid beta and alpha-synuclein. So with this in mind, one of the overarching questions in our lab is how do microglia become dysfunctional in chronic inflammation? We know that in early stages of Alzheimer's disease, microglia are able to take up amyloid, but in late disease stages, they're unable to effectively clear out the amyloid plaques from their environment. And secondly, we want to ask, what are the specific changes in the molecules that are associated with the diverse phenotypes in microglia in different conditions? Several gene expression studies, such as single-cell transcriptomics, have already identified different subsets of microglia in disease and in different conditions. But we are particularly interested in looking at the lipids and metabolites in microglia, specifically because these are functional molecules that directly affect protein function or signaling events within the cells. So, for example, lipids act as ligands for TREM2 and other receptors on microglial cell surface, dictating their downstream signaling events. And a class of lipid called as phosphatidylserines also, when they are exposed on the external cell membrane, acts as eat-me signals for microglia to prune neuronal synapses if they're expressed on neurons, or also on apoptotic cells. They engulf the dying cells when phosphatidylserines are expressed on the membranes. So this is a typical structure of a phosphatidylserine. It has two fatty acid chains and a serine in the glycerol head. However, this is just one class of lipid, right? Diving into some chemistry, we can actually appreciate that there are indeed many different lipid classes. For example, these are just a few that I have shown on the slide. For example, phospholipids. There are several different classes of phospholipids based on the functional groups in this R2 position here. So there's phosphatidylinositols, phosphatidylcholines, phosphatidylserines, ethanolamines, and glycerols. And also sphingolipids, for example, ceramides that are primarily on neurons, are also shown here. They have a long fatty acid chain with functional groups. Cholesterol esters, of course, cholesterol is huge for microglial function and astrocyte function. And also another neutral lipid, that's triglycerols, which have 3 fatty acid chains linked to the glycerol head. And what we should appreciate in the lipid classes is that the there are several different species of lipids within each class. In fact, there are hundreds of species based on the length of the carbon chain or the position of the double bond, and whether there is a double bond or not, that dictates whether it's a saturated lipid or unsaturated lipid, and the functional groups. And each have their own structure, and they influence different functions in the cell. So with that in mind, we asked, how can we profile the lipid landscape in microglia, lipid and metabolite landscape in microglia? And specifically, I asked what lipids and metabolites are uniquely made by microglia with amyloid beta. So I isolated primary microglia from perfused brains and cultured them in take media that was established by Barris Lab. And I treated the microglia with amyloid beta, and we isolated lipids and metabolites from the cell pellets and also from the conditioned media. And using a very sensitive mass spec technique called as MRM profiling, or multiple reaction monitoring profiling, that's a mouthful, we were able to identify different species belonging to different classes of microglia. In fact, we screened around 1,400 different lipid species that were organized into 11 different lipid classes. And we also, we have established this method quite well in our lab, and we also have bioinformatics pipeline to analyze these large data sets. And this experiment gave us large data sets that I have briefly summarized here. So the first thing I saw was what were the changes in different classes of lipids with amyloid beta over time. So here we can see that with initial exposures, direct exposures to A beta ex vivo, we can see phosphatidylcholines and sphingomyelins that are abundant in the cells, but over time, they disappear. And there are other classes that we can look at and ask specific questions. But more interestingly, we see here that with initial exposure to amyloid beta, we see abundance of free fatty acids in the cells. However, over time, there's a switch between free fatty acids and triacylglycerols, or tags, in the cells. And this relationship between free fatty acids and tags is an interesting one, because excess accumulation of free fatty acids can in fact be toxic to the cells. So cells have a way of protecting themselves from this overload of free fatty acids by performing esterification, which is a chemical reaction, and which thereby neutralizes the free fatty acids into tags. So this is a kind of metabolic reprogramming by the cells that makes themselves protective with increased exposure to A beta, at least ex vivo. So it would be interesting to translate this to in vivo, where we can extrapolate some of these lipid classes and look at the changes occurring within the microglial lipidome. And further, we also looked at some of the common lipids and metabolites that were expressed with amyloid beta treatment. So phosphatidylglycerols, for example, with 20 and 24 carbon chain lengths, which were saturated, were consistently upregulated in microglia. And at the same time, some of these long chain free fatty acids were consistently downregulated. And per the metabolites, there were tyrosines and tryptophans and asparaginins that were consistently downregulated. Now, what this all means, we're still trying to, ask fundamental questions about what these lipids are doing in microglia and what are their functions, and how can we validate these lipids functionally in the cells. So looking at metabolites, we can also look at what pathways are affected with the significant metabolites. So which are shown here, some of the significant pathways are highlighted here. For example, alanine and aspartate and glutamate metabolism, is one of the significant affected pathways. Arginine biosynthesis pathways are also affected, as shown on the right. So all the metabolites that are involved in this pathway were identified with our MRM profiling mass spec technique. So this this gives rise to a lot of interesting biological questions that we can follow up with. So with this technique in mind, we next asked, what are the lipids and metabolites that are unique to amyloid beta specific glial subsets? In other words, there are phagocytic glia and there's non-phagocytic glia. So what are the differences in the lipid profiles within these glial subsets? And before we could answer that question, we needed to be able to specifically look at these A beta specific glial subsets. So we synthesized a pH-sensitive fluorogenic amyloid beta human amyloid beta analog called pH-A beta to identify A beta specific phagocytic glial cells in live glia and in live tissues. So as we see here, when the reporter is taken up by the glia, there's an increase in green fluorescence. And one of the good things about this is we can use this for live cell imaging without the need for an antibody. And our collaborators, so Nils in David Atwa's lab has actually used this reporter to show the amyloid beta uptake by cells in a live mouse in real time in vivo. So, so as seen here, by creating a cranial window and applying our pH-A beta reporter, we see an increased uptake of the A beta by cells in real time in vivo. So to our understanding, this is the first time that A beta uptake has been visualized in live animals in vivo in real time. So using this, we also studied A beta uptake in various different animal models. For example, Pablo, again in David Atwa's lab, has used the reporter on acute hippocampus slices and showed that microglia take up more. I'm sorry, I can hear someone speak. I'm sorry. And he's shown that A beta is taken up more by microglia compared to astrocytes. And Emilia from Gottfisch's lab has used the pH-A beta reporter and showed by performing stereotaxic injection of the reporter, again, over time, that microglia and astrocytes both take up A beta. However, over time, astrocytes lose the capacity to take up A beta in vivo. And again, in another model where the A beta was injected into the retina, performed by Indigo from Shane's lab, we have again characterized the uptake of the reporter in microglia and astrocytes. And Nils, after he visualized the uptake in vivo, he could actually fix the tissues and then stain for different cellular markers, and the fluorescence was retained, and we could actually see the localization of the A beta in different cell types, mainly microglia and astrocytes and CD68 positive cells. So with this tool now in hand, we are able to, the data that was not shown is we can sort the cells. So please feel free to look at our paper that's on bioRxiv. So we are able to sort the cells and now look at the molecular changes within the phagocytic and the non-phagocytic glial cells. So in my talk today, I have shown for the first time the changes in lipids and metabolites that are in microglia that are occurring due to amyloid beta direct exposure. And I have also demonstrated the effectiveness of using multiple reaction monitoring profiling, which is a sensitive mass spec technique to characterize the cellular metabolome and lipidome. And we have also demonstrated with all our collaborators that the pH-dependent conjugate of A beta can be asked to look at A beta specific phagocytosis in vivo in glial cells. So I should mention that all of our tools and techniques are cell type independent. I am partial to microglia, but we would love to collaborate with anyone interested in using these methods and these tools in your own cell models. And ultimately, our goal is to, in question of our lab is to ask, how do microglia become dysfunctional in chronic inflammation? And we are going to answer that question by looking at different functional molecules that are expressed that directly influence cell function in vivo. So with that, I would like to thank first and foremost my advisor, Gaurav Chopra. This is us in 2018 after attending our first Glia meeting as newbies to the field. And I would also thank my thesis committee, Dr. Bruce Lamb, Kavita Shah, and Chris Roche, members of the lab, John Krupal, my undergrad mentees, Gabby and Liz. And I would like to plug in my LabMates poster number 63. So please talk to us in the Slack channel. Christina for helping with mass spec. And of course, our wonderful collaborators, Nils and Pablo from David Atwood's lab, Kevin and Indigo and Shane, and Emilia in Garfish's lab. And thanks to our funding agencies, and I would be happy to take any questions.
All right, thank you. That was fantastic talk. So we got time for a few questions. This one's from Catherine Askew. She's a postdoc at Edinburgh, and she says, it's interesting to see the changes in lipid profiles over time. Could it be that cells that can't metabolize FFAs into TAGs become dysfunctional and contribute to pathology?
Wow, yeah, that's a great question. In fact, that's what we are going after, like what lipids are directly influencing microglial loss of function. So that would be interesting to follow up. Finding inhibitors for lipids, actually validating lipids as functional markers is a very challenging question because there are so many subtypes and species and isomers of lipids. So we're actively working on that and trying to answer that question.
All right, another question is from a postdoc, Glia Albertini. Can your findings be extrapolated to other phagocytic contexts, such as development?
Yeah, of course. So as long as it's substrate specific, so I today focused on amyloid beta, but we can make similar conjugates using different chemistries with other neuronal substrates like myelin or tau proteins. So if you are interested in asking specific questions of phagocytosis during development, we can do that.
All right, so here's a question from Matthew Mold, a postdoc. Was A beta 42 phagocytosed in your CD11B positive microglia cell cultures? And did this affect cell viability?
Cell viability? No, because we optimized the concentration to be used that would not kill all the cells. So no.
Okay, here's another question from a student. Thuvan Liu. Have you looked at the uptake of different A beta states or length using your pH sensitive reporter?
No, we have not. So our reporter is very specific to the A beta 42, the human protein. So unless we make these reporters with different species of A beta, like oligomers, which is again a hard chemistry question, but that would be interesting to explore, but we have not done that.
Yeah, this will be the last question from Travis Faust, who's a postdoc. He's curious if you have an idea as to whether the change in lipid profile over time is due to changes in the microglial metabolism transcriptome versus different rates of engulfment or degradation of different types of lipid. That's a good question.
Yeah, so we have not looked at transcriptomes. So I don't have an answer for that question, but the second part, which was about the changes in function, I believe. So that could be, so we don't know if the function is influencing changes in lipid profiles or if the changes in lipid profiles are influencing function. So again, that comes back to, validating lipids as functional markers, which is a very, very hard problem, but that makes it even more challenging. So I do not have a direct answer for that question, unfortunately.
That makes it interesting. All right, so thank you for a great talk. I think we're on to the next talk.